Our next tissue in mammalian histology is nervous tissue. This tissue has a power of responding to the stimuli or disturbing influences such as may be produced by mechanical forces, sound, heat, cold, light, chemical actions, and electricity. Protoplasm also has the property of conductivity, by means of which impulses set up by stimuli are transmitted from one portion of protoplasmic mass to another. In the cell, which make up nerve tissue, the properties of irritability and conductivity are more highly developed than in any other tissues within the body. The nerve system is made up of three components: neurons, nerve fibers, and neuroglia. Here, this slide shows anatomical structure of neuron. Neuron is a structural unit of nerve tissue. Together with their processes, the neurons develop from the neuroblast cells of the embryo. Each neuron is made up of three parts. First part is the cell body or cyton or perikaryon. This portion. Second part is the dendrites, the branches to the main body. And third one is the axon, the part which is embedded in a peripheral nerve system and a body. This cell body or cyton, a part of a cell in which the nucleus lies, is called the cell body. The nucleus is large and spherical, contains a single distinct nucleolus. In the cytoplasm, mitochondria, Golgi complex, fat granules, and neurofibrils are seen. The centrosome is absent, so that the neurons cannot divide, but according to some authors, centrosomes remain present in neurons. Beside neurons, basophilic bodies are found in cytoplasm. They are called Nessels bodies or granules, which here colored blue. These are made up of a chromatin. The significance of Nessels granules is not understood, but their importance in the metabolism of neuron is indicated by the very definite changes which they undergo under the varying physiological conditions. Nessels bodies are found in dendrites but absent in axon. Certain flask-shaped cytons, Purkinje cells, found in cerebellum. Second part of a neuron is a dendrite. They are usually numerous, many, which are short process showing a high degree of branching close to the cyton. These processes connect neurons with the others. So this central neuron cyton is connected with each other through the dendrites. This axon is a single process. It is usually given off from a Nessels granules, free small conical regions on a cell body, which is known as an axon hillock. This neck portion produce axon is called as a axon hillock or implantation cone. The cone contains ribonucleic acid, protein, iron. The axon ending or terminal arborizations are telodendrites at its free end. Collateral branches may be given off by the axon along its course, but these are frequently lacking. The cytoplasm is known as exoplasm, which lacks Nessels granules, Golgi complex, but neurofibrils are present. Here in these stained slides, we can clearly see a structure, cyton structure in this first slide. Here at the centrally located nucleus is there, nucleolus is there, then nucleus is there. Then these black spot, they indicate the Nessels substance, which are chromatin substance, and these three ends indicate the dendrite processes, and a single end here is a axon. Now in these dendritic spines, when stained with the silver, it shows this exodendrite synapses, and this dark color portion is a main body of a nerve cell which produce these dendrites, and they are joined with each other and they form axon at the terminal end. Types of neuron. A great number of types of neurons may be observed in various parts of nervous system, like unipolar neuron, pseudo-unipolar neurons, bipolar neurons, multipolar neurons. Among that, we'll see first this unipolar neuron. The unipolar neurons are those in which only a single process, the axon, passes from cyton and no dendrites are present. These neurons are rarely found in the central nervous system of adult, but they are present during developmental stages in early embryonic life. Pseudo-unipolar neuron. Here, these are a typical bipolar cells which are found in all spinal ganglia and in the ganglia of a cranial nerves other than eighth nerve. These cells are first typically bipolar and spindle shaped, but as development proceeds, the processes converge until they meet at one side of the body. These then elongate so that a fine process is formed with the T-shaped division at the end. One branch of T being dendrites and other axon. Now bipolar neurons. Typically bipolar neuron has a single dendrite and a single axon projecting from opposite ends of the cell. Such neurons are present in the retina of eye, in the olfactory epithelium, and in the ganglia of eighth cranial nerve. Now multipolar neurons. These are most numerous of all, is variable and depends upon the number, arrangement and length of processes. They may be two types. In Golgi type 1, there is a single long axon and numerous short dendrites. In Golgi type 2, the axon is short. The cyton of multipolar neurons are located in the gray matter of brain and spinal cord. In Golgi type 2, the cells are confined to central nervous system, but the axons of many neurons of Golgi type 1 forms the motor components of spinal nerves and of certain cranial nerves. Neuroglia beside nerve cells and vascular tissue. Here in this diagrammatic presentation of nerve cell body, you can see an entire nerve cell from axon. If we start studying this diagram, then centrally located axon is covered with a mesoxon. All these axons, they have vesicles or neurotransmitters and a fiber-like structure, that is neurofilaments. They continue with the neurotubules, and this is a portion called axon hillock which joined with the main body of nerve tissue, nerve cell, which joins with the main body of nerve. In the central body of nerve cell, there is a centrally located nucleus, cell organelles, Golgi bodies, mitochondria, and dendrites of another neurons are joined with this neuron through exosomatic synapse. Here exodendritic synapse are joined with this main cyton body of nerve cell. In this gap you can see a axon of another neuron passing and surrounded with the myelinated sheath. In this case, There are axons, but they are surrounded with unmyelinated sheath. In this photograph, we can see a presence of dendrite here, a long whitish channel-like structure that represent the dendrites. And here, this round portion, dark color round portion around the white part is a myelinated axon. So centrally white color portion is axon and this is a myelinated sheath. In this diagram, this is a brain, and from the brain originate these nerve cells, neurons, cell bodies are there, and this red color axon transfer or travel through the spinal cord organs. Now these ganglions are these neurons are Golgi type 1 neurons, and when they reach in the center of this spinal cord, these are the Golgi type 1 neuron present in a gray matter. These Golgi type 2 neurons are present here, and in a white matter axon tracks are present. In this photograph, this is a diagrammatic presentation and this is an actual color photograph of a slide showing neurons in a cerebral cortex.
Neuroglia. Besides nerve cells and vascular tissue, there are found certain cells whose nuclei do not possess any nucleoli. These cells constitute neuroglia cells or simply the glia. In the central nervous system, there exists a ratio of 10:1 between neuroglia cells and neurons. The neuroglia cells may be divided into different classes as astrocytes or astroglia. These are star-shaped cells with many branching processes. They may be divided into protoplasmic astrocytes and fibrous astrocytes. Protoplasmic astrocytes are mainly found in the gray matter of brain and spinal cord. The nucleus is oval in shape. The cytoplasm shows the presence of small rounded, somewhat elongated granules called gliosomes. These may be considered as counterparts of mitochondria. Some processes of protoplasmic astrocytes are attached to the walls of blood vessels by means of expanded vascular end feet called foot plates or perivascular feet. Microglia or mesoglia, also named as oligocells. These cells are mesenchymal origin and appears in the central nervous system only after the blood vessels have invaded it. Microglia cells are small cells with a narrow, elongated nucleus each. The cytoplasm is scanty and often drawn out at opposite poles of cells. They are found both in a gray and white matter, but more in a white matter. The macroglia cells act as brain macrophages and possess the ability to phagocytosis. Sometimes they become actively amoeboid and phagocytic. They swell up and known as beta cells. These phagocytic cells are included as a part of reticuloendothelial cells. Oligodendrocytes or oligodendroglia. These are the commonest type of neuroglia. The name was given because they contain branches which are small and fewer as compared with astrocytes. Cytoplasm is scanty and more dense. The white matter they unsheath nerve fiber to form myelin sheath. In the grey matter, oligodendrocytes serve as satellites to the cytons of neurons.